we are going to dig into section 2. Okay, and so in section 2, things start to get more, I think it's been interesting so far, but I think it starts to get a bit more exciting. Okay. So let me just share screen. Let's get rid of that. Okay, so this is where we actually get differentials. This is where we get to understand which genes are changing, which ones we care about, essentially. And so, just make this bigger. And so this is the session 2. And just while we have this up here, this will be the final session for today. We're going to go from 3 until 4. Optimistically, as it's already 3:10. Maybe we'll overrun again. I apologize. And then I'll give you some time to do the exercises if anyone wants to stick around. And then I'll run through the exercises. And everything will be done by 5. Tomorrow, starting at 1, tomorrow's, I think, is the really exciting stuff, because as soon as we get to know what genes are changing, you want to know what is the relationship between these genes, what is the functional relevance of that, and that's what we're going to start to cover tomorrow. And so we'll have a whole section about how to visualize these, how to understand how they relate to each other through using things like PCA and clustering, how to work with multiple data sets as well. And then we're going to talk about gene set analysis. So this includes Go terms, GSEA, and things like that, KEGG. And this is typically one of the first things we get asked to run from people that come and work with us. And so tomorrow's going to have some really exciting stuff. But to be able to do that, we need to actually understand which genes are changing and what's the relative, like log twofold changes and things like that between my conditions. So we'll be using the same data, okay? But now we're going to start considering, well, not just one replicate at a time, we need to consider multiple replicates, and we need multiple conditions. We're going to be in exactly the same place, so that hasn't changed. And so the big thing here is we've done a lot of this work for you. And so I feel like some of the thing that's kind of held, maybe, I know a lot of people like code along while we talk through this. One of the things that's kind of held you back from doing that is some of this has been really big. You can't build indexes on the fly. It takes a lot of time. The step we're at now, once you have counts, it's super straightforward, okay? And it's much quicker. The longest steps take 30 seconds as opposed to 5 hours, okay? And so all of the data objects that contain the counts are available in the, as long as you have your R in the right directory, okay? And we have the counts in genes, which is in the gene counts.r data. We have the disjoint exons, we won't actually get into today, I don't think. And we have the results from salmon. And we'll do a little comparison between salmon and working with salmon and working with summarized overlaps. And you don't have to load them in right now. I'm just letting you know so you know where they are. Okay. So as I mentioned, we're not just going to be considering one data set, we're going to be considering many data sets, right? And so if we're going to do this, as I mentioned earlier today, you're going to want to be able to count over multiple data sets at once. If you're working with salmon, that's super straightforward. You can just tell it where multiple BAMs are, or multiple FASTQ files are, and it will give you multiple outputs. Within summarized overlaps, I did kind of mention it or alluded to it earlier as well. We can do exactly the same thing in the sense that we can have a vector of BAMs that we care about. And so in this case, we have this vector here. And so in this analysis, we're actually, we're going to do a 2 versus 3 comparison. So we have 2 Treg data sets and we have 3 activated Treg. And so we have the position of these 5 BAM files, and we're going to say them into a vector. We give them names. So this is just a better name. It's exactly the same pretty much. It's just drop the .bam. And now we have this BAM file, BAM files to count object. And instead of last time we just used BAM file function, we now use a BAM file list function because we're giving it more than one BAM file. Okay. And so now we've set up the specifications of our BAM files. We give it an appropriate yield size. So again, this is so you only read in a certain amount of reads at once instead of all of the reads. And this can help with your memory. And then we can just run summarize overlaps in exactly the same way. Okay, so we get this G.exon object and we run summarize overlaps. And the big thing that's changed since we did run it earlier today was my BAMs is now a BAM file list with multiple BAMs. Okay. And so if we can see that in the result, in the dimensions, it's changed. It's now 5, it's now got 5 columns. And you can see that the column names correspond to each of these replicates. And so something we've kind of alluded to and talked a little bit about is I've had lots of questions about running this on the HPC. Okay. And so we don't go into tons and tons of details about how to optimize this on the HPC, but the working in environments like that, there is a package that's very, very useful, and it's called Bioc Parallel. Okay. And so when you install this, it enables you to do things like parallelization. Okay. And so what do I mean by that? Well, instead, if I run this as is on your computer, it will send out one job and it will just count in one file. Then when it finishes, it will move on to the next file and count. If you're working on a HPC, what you can do if you have Bioc Parallel set up correctly is you can get it sent out and it'll count on all 5 files at once and it will speed things up. And so that's the benefit of using something like a HPC. And apart from like it having more memory, is you can actually push it a little bit further. And so the way it works is there's these different param setups. So you can do a multi-core param or a serial param. Serial means you're just using one and you're doing one thing at a time. Multi-core param means you're setting the specification so it's using multiple. Okay. And so again, you can set up these parameters and then you register those parameters. In this case, I didn't want to change what we have here in this R session that I'm using to build the slides. So I re-registered param serial. Okay, but you could do param multi and then do register param multi. But read into it, all depends on like what your setup is, how many things that you're trying to do, and how confident you are in the HPC. But you can read into this package, Bioc Parallel. That will have a lot of help, and the folks at HPC will also give you a lot of help as well. So work is equals 2. That means I want two jobs, essentially. So instead of doing it once, I'm going to do 2 things at once. So in my case of 5 samples, right, it's going to send off, it's going to do file 1 and file 2. And then when it finishes the next one, it will do file 3 and file 2. And then when file 2 finishes, it will do file 3 and file 4 simultaneously. It's this idea of multiple jobs at once working in parallel, as opposed to 1, 2, 3, 4, 5. Okay, so we only kind of touch on that. I know you guys are interested in more. We're just not necessarily the right people for it. Okay, so next up is having a look at the result. So you can load in this counts object here. So this is our gene counts. It's an RData object. You should be able to just load it in. And if you load it in, you can access it with the assay function. And so if we did this kind of earlier, but these data objects that are a range summarized experiment, they have specific ways to get into them and to break them apart and then to work with them. And the assay is one of the most important functions. And it's just saying, I know you have all this information in it. I just want to look at the counts. I want to look at the result of whatever this experiment is. And when it does that, you can see on the left-hand side as the row names, you have the gene IDs, and then along the top, you have the col names, which are the column names, which these correspond to your sample names. Okay, and so you can see that we have most of these. We have 1 in this case. It's pretty low counts. This is all the way up at 11, but that's still pretty low counts. Okay, so we have our count table now. The other things that you can do with this object that's stored within it, it remembers the ranges. So this is really nice if you want to figure out like where is my gene. You have this ID, right? You have this ID, but that doesn't, like, I could figure out what gene that is if I wanted to, but stored within the actual object, the counts object, or this range summarized experiment, is actually useful information. So if I use the row ranges function, and again, I'm looking at the first 2 rows here. I'm just subsetting it so I can just look at the first 2 rows. What I get out is a granges object. So these are another key bioconductor object, and they have all the information about the exons associated with this specific gene. And so by using these summarized experiments, you can contain a lot of information in a single object. And they're all related, and you can use like intuitive things to start to access them. So what I've shown you is this assay for getting the count information or the result of the experiment, and row ranges to get the ranges or the regions of the genome that are associated with that specific line or row. And so now you have all this information, you know about the gene, you know about what it contains, where it is, you can actually start to do some differential gene expression analysis. And so to do this, there's a bunch of like steps involved, but and so some of these steps are like, you want to transform your data, you need to normalize the counts, and then you need to calculate the significance between your different groups from these normalized values. And there's one package that kind of takes care of all of this, okay? And that is Dsq, okay? And Dsq has a specific like favorite way of doing these normalizations. It builds models based on what the data actually looks like. Yeah. And so, yeah, like it says here, there's 2 main different, like Dsq has a specific way. There are other tools that use different methodologies where you actually do like log2 transformation first, you do a bunch of processing first, and then you do your differential analysis using standard tests and normal distributions and things like that. But Dsq just wants the counts. Dsq just wants count information, and then it's going to use distributions which are more intuitive and much closer to real data. And so first step really involved is, again, this is going to be taken care of by Dsq, is the normalization. And so what we mean by normalization is this concept of scaling factors, or how many reads did you sequence per sample. So if you sequence 1 million reads in one sample and 500,000 reads in another sample, the counts per gene are going to naturally vary because you've just sequenced deeper. And so you need to normalize for that. And this kind of library composition that you have. So there are simple metrics for doing that, like counts per million or some reads per million, depending on what nomenclature you use. But more sophisticated methods like Dsq, they find these scaling factors, and then they use those scaling factors within the models that they build. And so I've been mentioning a lot about models, and what do I mean by models? What does it matter? Well, typically in an RNA-seq experiment, you're doing quite often 3 replicates per sample, and then you have 20,000 genes or something. And so you have this multiple testing issue combined with not that many replicates. And so 3, often people say that's enough replicates. If you were to like get deep in statistics, it's really not very many replicates at all. And so to get statistical significance, what is often done is models are used to try and help minimize the variation. And so what we mean by that is between your 3 samples, you want to minimize the variation so that when you can compare group A to group B, they're as different from each other as possible for your statistical test. And so by helping to minimize the variation, that can help allow you to call more genes as significant and find regions which actually are changing. And so one of the key ways to do that is there is a relationship between your average gene expression and the amount of variation you have. And you can kind of see that here. And so in this plot, you have a lot of variation relative to the gene expression, while higher genes or more highly expressed genes have lower variation. And so by building a model of this, you can then help squash some of this variation, especially at the lower end, to give you better power and strength to call significance. And so, as I mentioned, we're going to be using Dsq for this. There are other tools, and none of them are really invalid. We sometimes lean on some of the other tools depending on the approaches we're doing, but we like Dsq. It does a really great job. Both EdgeR and Dsq work in very similar ways. They have count data that goes in. So what we take our range summarized experiment and we do assay, that's sufficient normally. While VUM and Lima, they do a log2 transformation first. And so that can it's a subtle difference in how the inputs are made. Okay, so before we get into the nitty-gritty of this like normalization, which I was touching on, and this like variance shrinkage, first we need to set up for Dsq. And so we could just give it the counts, but we also need to provide it with information about what those counts mean, what samples belong to which groups, and things like that. And so we're going to go through this workflow. And the first step is giving it this sample information. And so this sample information is conveyed by a data frame. And your data frame should only have one group or one column, and that will have the group information. And then it will have some row names, and these will be the names that are in the count object that you have, and they'll be the column names. Okay, and so we can use the data frame function to set up this metadata. And so you can see what this looks like. When we do this, we'll have one column, okay, and we have their group names is naive or activated, and we have what that corresponds to, okay? And so if we remember back, like here, we're now just marrying up this group information of whatever we want this group name to be with these count table. So we know that these two are part of the naive group and these 3 are part of the activated group. Okay. And so then we can also, and the other parts we need for Dsq is we need the counts. So we can just do assay and grab the counts out. So that's the object we were just looking at. And we can get the row ranges, okay? You don't have to provide the row ranges, but it's nice because then all future, like in the result object you create, it will still have the ranges associated with it. So when you look at the results, you can then go, oh, where is this gene ID? Like, where is it? I have the ranges, I can understand it, I can look at it. Rather than having to cross-reference back to a previous object. And so then you put all this together in one function called DESeq dataset from matrix. And so what this is doing is it's setting up your Dsq object. Okay, and if anyone uses Dsq in the future or needs help with it, the Dsq help page is amazing. The guy who runs Dsq and maintains it is very, very responsive. And you'll find in these help pages, everyone calls this object a DDS object because it's a Dsq dataset object. Okay, so you'll see that everyone uses this nomenclature as DDS. It can be whatever you want it to be. It doesn't have to be DDS, but it just makes life easier because everyone calls it DDS. And so we provide it with the count matrix. We provide it with the metadata, which we created. We provide it with the row ranges. And then most importantly, we provide it with the design, okay? And so our metadata has only one piece of information, right? It's a group. Is it part of activated or naive? You can have much more complicated setups, much more complicated experiments, where you'll need a more complicated model. Maybe you have multiple groups that you're comparing. Maybe you have wild type and mutant, but also drug and treatment. And so you have 4 groups and there's a relationship between all of them. There are specific models you can use to help understand that and that relationship. But for us, for now, we'll just use group because we want to compare activated to naive. Okay, and so once we do this, we have a Dsq dataset object. It contains all this information, but it hasn't done anything to it. It's just all there, and it hasn't done any actual analysis with it. Okay. And so, yeah, when you're setting this up, if you don't want to have to provide this cold data information like this, these objects, this DDS object here, and this range summarized experiment, which contains your counts, are very, very similar. And so you can actually add the group information directly to the count information here. And so you can see it now has a cold data here, which is group. And then when you run Dsq dataset from matrix, you won't need to provide it again because it will already be there. So in this case, you can just do this and you can simplify Dsq dataset. Okay, and it will build it directly from your range summarized experiment. This is kind of designed in a very bioconductor way where it knows you're pretty much getting it from a range summarized experiment, and it takes as much information as it can from it as possible. So this is just an alternative. Okay, so that's 2 ways of going about the same thing of setting up your Dsq dataset object. And so now you want to run the Dsq workflow, okay? And so there's one function that will do everything for you. And you can see when you run it, it goes through these steps. Now, this is probably the longest process we'll do. It takes about 30 seconds, maybe, depending on your computer. It might be quicker, it might be slower. And all we do is do DDS, we give it to the Dsq function, and then we assign it back to DDS. And it does all these steps. It estimates the size factors, dispersion, it makes some models, and it does some tests, okay? And so we can actually break that down a little bit so you can understand some of these steps. And so, for example, when it does estimating size factors and starts to calculate normalized values, we can extract those out. So if you use the counts function on your DDS object, you can ask it for normalized equals true. And so when you do this counts function, what it will return is a counts object where all the values have been normalized. Okay, and so what I mean by normalized here is they're normalizing to read depth. So it accounts for like your counts per million and things like that. You can also do normalized equals false if you want to get back your original count table. So everything that was available in your original count object is still available in the DDS object. You just need specific accessors to get it out. And so getting to the variance estimation, which I think is one of the most interesting parts of this, and what I kind of talked about earlier, is DESeq, the strength of DESeq, and some of these specific methods which are completely designed for doing RNA-seq differential gene expression analysis, is the strength is in the shrinking of the variance. And so by shrinking this variance, it becomes a lot easier to call significance between genes. And it's using this modeling information to be able to do that. And so by understanding the fact that, hey, at some of these levels, we kind of expect this amount of variation, we can actually make it a little bit smaller because this fits with what we expect. And if something has more variation than you expect, then maybe we won't shrink it because that's probably real. And so this is, you can use the function plot dispests to get an understanding of what that means and how that looks for your data set. So when we do it for this data set, you can see that the black dots are the gene count estimates, okay? And so that's the kind of the estimated amount of for your data coming in. It's been normalized. This is the expected number of counts on the bottom, and then the dispersion level. And so we can see that, again, this relationship between low counts, high dispersion, high variance. Then we have, in red, we have this fitted line. So this is the relationship or the model between the counts and the variance or dispersion, okay? And so then you can shrink towards that model. And so the final value are the ones shown in blue. And so everything in blue shows the kind of final values after they've been shrunk. And so on the bottom, like you can see how all these black values have been brought inwards. All these black values down here have been brought inwards. And so the dispersion values are reduced and much more similar to each other. And then you can then see up here, these values which were more considered outliers have not been shrunk. So it's bringing everything into a closer register with one another, which allows better testing, but it's not doing it for genes that already look weird or already look like they do have very high variance. Okay, it's only doing it for ones where it makes sense for. And yeah, so if is there, so that makes, I think this makes sense if the ones above, so we have a question, why is it only against the ones below, and the ones above and not below that are not being shrunken? And so these are the ones with very high variance, right? So we have this, the variance is on, or dispersion is on the axis. And so these are the ones we're concerned about anyway, is the ones that are high of the line. They're the ones that are anomalous. If we're bringing stuff upwards, we're not too worried about that creating something like a biological artifact. But these ones, if we brought them in, it would be a problematic. Okay, so those are the two main steps. We have the normalization step, and you can extract out those normalized counts if you want to be able to do stuff with them. And then we have this variance step. Okay, all of it is taken care of in the DESeq function. Okay, so you don't have to look at those two. You don't have to look at the counts. You don't have to look at the dispersion estimates if you don't want to, but they're there in case you want to. Okay. What we really want to be able to do is actually get the comparison. Okay, so when you do the DESeq function, what it's doing is it's interrogating your data, it's setting up these models, it's doing the normalization, but it's not doing the pairwise comparison. And so to do the pairwise comparison, there's a separate function called results. Okay, and so in the results, when we do a results function, we say we want to do results, we want to do it on our DDS object, we want to do a specific contrast, and we want to consider the group information, and we want to compare activated versus naive. And the ordering of this does matter. It won't change your result. You'll still get the same significant genes coming out, but this is essentially the order if you were to consider like a log twofold change. And so you typically will want to put your mutant, your condition B, your treatment first, and then you'll want to put whatever control or wild type is second. Just like in the log twofold change, if you have a log twofold change on the top, the numerator would be the mutant, or it would be the the treatment or something like that. Because then when you do the comparison, if it's a high value, that means it's increased in this treatment or this mutant versus the control. So that's what the ordering. It doesn't necessarily change the result, but for ease of interpretation for everyone else, whatever your baseline is needs to be second. This is a typical step we do is what this produces is a special results object. And you can see this is the object at the bottom. But before we look at it, we typically organize it so that it's arranged by either p-value or adjusted p-value. So we have the most interesting things at the top. And so what we'll do is we use the order function, and we do order on myres p-value. And what this does is it creates a vector, a numeric vector, that is listing out which has the lowest p-value. And then that was number 1. And then which has the second lowest p-value, that's number 2. And we put that into myres to order the indexing. And so then that will reorder my res, or my result from this, into having the most significant first. Yes, so we just had a question about correction for false discovery rate. Yes, and so this is inbuilt. And so I'm going to get into false discovery rate in a sec. But so we will get to it. But you can see here we have the adjusted p-value. Okay. And so in this result, I'll just quickly go through the columns. We have base mean. So base mean basically is the average gene or whatever thing you're looking at. But in this case, average gene counts across all experiments or all samples. And so this gives you an idea of like, is this a highly expressed gene? Is it a lowly expressed gene? Obviously, if you have it crazy high expressed in one sample and none of it in another sample, it will still be quite high potentially versus other ones. But this is still quite informative. We then have the log twofold change. We have the standard error associated with it and the statistic, then the p-value and the adjusted p-value. So this is the one that's corrected for FDR. And we'll get into that in a sec. Yeah, these are all written out here. Okay, for reference, but we just went through them. And so Dsq has some nice functions, so we can use like a summary function here, and it'll tell you a summary of the result. And so in this case, it's telling you how many of a certain p-value are up or down, and how many have outliers, or how many have very low counts. And so you can straight away get an intuition, like from this, you can see, oh, these are my genes that are changing the most, right? And then through this, you can get a much more global view of what's happening, which is nice. So using those two in tandem, once you get your result, you can actually start to investigate what's happening to my genes. Do I have a global change? Is there a bias? Is everything going, all the ones that are changing, are they all going down? That kind of thing. Quite a nice tool to also be able to visualize these changes is an MA plot. Okay, and so what MA plot shows you is the mean of like the counts essentially on the x-axis, and the y-axis it shows you the log twofold change. And then if you're using the plot MA function, which is part of Dsq, you do that directly on your result object here. And the nice thing it does is it highlights in blue everything that's significant. And you will notice that there is a relationship between significance and the number of counts, right? And that makes sense because lower counts means it's harder to call significance. And even if we do things like our variance estimation, there's still, that helps, but it's not going to fix it. A sec. Something we can do is downweight genes with high fold change but low significance. And so you have some like here where they're off the plot. You can see these little triangles, right? They're off the plot. They're the highest log two fold changes, but they have relatively low expression versus some of these ones up here. You get this kind of violin shape, right? And actually that can, to a certain extent, be less reliable. There is this relationship between lower significance because of your lower counts, but higher rates of log two-fold change. And often those are the regions you just don't care too much about. Like most of the time we care about regions up here where you have a high amount of counts, it's robust, the change, and it's significant. And so you can use something called LFC shrink, log fold change shrink, and you can do it on your DDS. Okay, and what this does is it penalizes against things that are low significance but high log two-fold change. And then to do this, you have to supply the coefficient. Okay, so in this case, we're saying it's similar to what we did when we did the results, right? In this, we're saying we want a contrast between group, activated versus naive. And so the coefficient for that is group underscore naive underscore verse underscore act. And so this is the coefficient is stored within the DDS object. You can extract them. You can have a look at which ones are available to you. But if you just follow this kind of naming pattern, you'll be able to also just substitute the names out and perform something similar. And then when you do the MA plot of this, you can see that it's less shaped like a like it's expanding out here. It's less shaped like a pear now than it was before. Like here it expands out, and these low log two-fold changes are penalized against. Okay. And so this is nice because it allows you to use values such as the log two-fold change. Like the log two-fold change at this point has some consideration of your significance. It's been scaled in some ways, and it has been augmented by the significance. So you can then at this point start to use it for things like GSEA. Before that, we wouldn't use it for GSEA. But after that, you could use it for GSEA. And we're going to talk about GSEA tomorrow. This special results object that we were looking at here, here, this object, as I mentioned, it's a special Bioconductor Dsq results object. Okay, but if you want to do things with it that are like regular R things, not bioinformatics R things, you can convert it to a data frame just by doing as.dataframe. And then it has a normal shape. You will lose the ability to do specialist functions on it, but you could always, in this case, we say myres as a df. We have a data frame version and a myres version of our result. Okay, Charles has a question. Do we usually use p-value or p-adjust if we would set up a threshold? So I think this is a perfect time to ask that question because we're about to talk about multiple testing. Okay, and so hopefully most of you know what multiple testing is. It is a really important step for when we consider genomics, especially, right? Because we're considering thousands and thousands of genes. We're doing many, many statistical tests. And if we have a 95% confidence interval, that means there's a 1 in 20 chance that we're wrong, which is fine most of the time. But then if we're considering like 20,000 genes, you're expecting to be wrong 1 in 20 times. So you're going to get hundreds of thousands of, well, say you do 20,000 genes, that's 1,000 genes you're expecting to get wrong, and that's your base level. Okay. And so looking at, yeah, we have to do some kind of correction to deal with this, but luckily there's many, many tools. And so, yeah, there's a cool comment here that you should read from XKCD about this. That's basically detailed. I'll just read through it real quick. It's basically detailing if you test a jelly bean color, they're testing jelly beans and they test 20 colors. And they're saying there's a correlation between jelly beans and acne. And they do it 20 times. And then one of the times they get really excited and it gets published that green jelly beans cause acne at 95% confidence. But it could have been any of them. It's just statistics at that point. And yeah, so there's multiple ways to do multiple testing. Typically, people talk about Bonferroni or Benjamin Hochberg. There's other types as well. But what we do typically is Benjamin Hochberg, and that is what is the p-adjusted value, okay? And the reason for that is this value, so to get the p-value, you multiply it in Bonferroni, the p-value by the number of genes you tested. So you're multiplying your p-value by, say, 20,000 genes. And so hardly anything is significant. Instead, if you do the same, but then you divide it by the ranking, it means that if you have a very significant, like your top most significant are going to be penalized against less than your bottom most significant. And so this is really nice because then it actually allows, it stratifies your data a little bit more. And the result of the p-value, and it actually allows you to hit significance in a much more reasonable way than this. This is idealized, it's an ideal scenario, but it's just not realistic. And so we can then do these multiple corrections and then apply a new cutoff based on that. And so typically what we do, I'd say, and I do, is I'd use the p-adjusted value. Okay, so that's your answer, Charles. It's the adjusted p-value is the one you're really interested in because it has accounted for that. When you go to thresholding, your most significant result should stay the same. Like whatever's at the top of the list will stay at the top of the list. It's just where you draw that line of it has reached significance, you'll get a different number as a result. And so we do independent filtering at that step to reduce the number of genes. And this is important because later things that we do, having a reduced number of genes can help give us more strength at that point. Plus, having less genes helps you focus on the needle in the haystack becomes smaller. And so a big thing that we need to strengthen and say here is you can't simply filter your differentially expressed genes and then do a correction. And then, yeah, you can't just do your filter at 0.05 and then reapply the correction afterwards, or rerun your differential expression. Okay, I'll repeat that because I think I muddled my words. So say you do your DESeq differential gene expression, you do a filtering of your adjusted p-value of 0.05. It has been known in the past that some people have then taken that result and then put it back through DESeq or put it back through differential gene expression analysis. And that is very bad practice because you're not at that point doing independent filtering. You're not doing anything that's good. And then they'll like reapply the correction afterwards. And that is not independent, right? Everything is dependent on those first steps. You're repeating the analysis in a cyclical manner. And it's bad practice, bad statistics, and yeah. It's normally only done out of desperation because there isn't much significant changes in their data set. And so they get sad. But that could just be the way that data set is. In your result from DESeq2, you may notice that it isn't just values. Once you've done your adjusted p-value, there is actually a bunch of NAs. Okay, and so in this case, we can ask the question, is NA? And we're specifically looking at the adjusted p-value of our data frame. And then we do table, because what table does is it counts everything up. And we can say that 10,000 are NAs and 13,000 aren't. So there's half of the result is NAs. What does that mean? Well, you can run your own p-adjusted value to compare to the one that DESeq does. And if you did this, what you'll see is that the ones that are NAs are typically would have an adjusted p-value of 1. Okay, so these are really weak p-values. They are definitely not significant changes. They are not genes you care about or are interested in. So why is DESeq making it NA? So if you were to look at, say, the so we don't actually explain it in the slides, but often there's NAs because what DESeq does is it wants to remove as many genes as possible before you do multiple testing. Because if you reduce your data set before multiple testing, that means that you can get more significant values. So genes it considers not worth counting prior to doing the multiple testing, it will remove. And typically these are ones that have very low count values to begin with. So what it does is it does a filtration step of things that have very low counts, and then it will do its adjusted p-value. So if it has an NA, that means it wasn't considered for multiple testing. Okay. And typically what we do in subsequent like downstream analysis is we'll filter those ones out. Okay, so like the first step is doing a filtration based on whether it's an NA. And then if you're only interested in significant genes, you can do a filtration step based on if it's past 0.05 or 0.01. And so to do that filtration, we just keep using this is.na function. And so you can do is.na, you ask, is any of this row or any of this column an NA? It tells you which ones they are by true and false. And then we use an exclamation mark. And so what this does is it flips whether a true or false is true or false. So it becomes false true instead of true false. And so when you're asking the question, is it an NA? The answer, if it was an NA, becomes false. And the ones that aren't NAs are true. And so then when you index with this true false, you'll keep anything that isn't an NA value. And so using this step, you can then filter, you can then organize by that. And then you can reorder. It shouldn't affect anything. You can order on p-value or adjusted p-value. You can then also, like, because a lot of these are zeros, you can add a second ordering operation. So quite often what I'd do is I'd order by adjusted p-value, and then by absolute log twofold change. So you have, if you get things that are zeros, then super significant, then it'll organize by log twofold change after that for all the ties. But yeah, you can see across the board, like the ones that are super significant, they haven't really changed. P-value to multiple testing, but doing it with everything as opposed to the filtered down version that Dsq does. Okay, so that is running Dsq, okay? And that's the way we typically do it. And at the result there, you have a data frame, okay? So I'm going to quickly show you how we do it with salmon. Okay, Charles had a quick question before we dive into that. How many biological replicates per group is recommended for DESeq2 analysis? So it depends on your experiment. For a typical RNA-seq experiment, we expect 3 and 3. In this case, we're using 2 and 3 just because I think one of the data files wasn't working, so we just dropped it out. An unbalanced experiment is less than perfect. You can run it on a 2 versus 2 if you want. DESeq will still function. But you will have cases, right, where say one gene is acting out in one case and you have super high variance and you don't know which is which, and it's hard to tell unless you have 3. If you want to deal with like if something goes wrong and stuff like that. So yeah, I'd say at least 3. If you are working with clinical data, it gets a little bit more complicated, especially with like, by clinical, I mean either human data or I guess it's not clinical. So human data or in vivo, like mice experiments, often you might need more than that because you have so much more variation. So if you have anything like behavioral studies, you might need bigger numbers, or if you have people that have genotypic variation, you'll need bigger numbers. And depending on what you're looking at, those numbers can swell to quite a lot more than 3. So it all varies, but your typical cells in a dish, and you're going to give one of them a drug, yeah, you can do 3, and that's the gold standard. Okay, so we'll dig into salmon. And so what we produced at the end of the last session was we produced basically a data frame, a file, which was an quant.sf file, which contained counts in it. And it's using this pseudo aligner, which is very quick. And so salmon, the people that made salmon and the people that make DESeq2 are actually very friendly with each other, and they've designed the 2 tools to work together in tandem. And so to do that, there is a linker, and that linker is tx import. And so that's another package called tx import, transcript import. And this works with other softwares as well. It isn't just for DESeq2 and salmon. It works for, I think it works for Edge. Does it work for EdgeR? It might work for EdgeR as well. Yeah. Oh, Sang asked, kind of random question, to create .band files, we use the subjunct function, but that seems the subjunct is not available in the current bioconductor. If that is the case, I should double check that. That would suck if it's not in there. It should have been tested. Should have been tested by our build. Let me just check. I have it on my. Hey, don't worry about interrupting the flow. We're about to start something new, so it's not too worrying. I'm already running late because I talk too much. So So it's all good. So I just opened up the Bioconductor page for subread on my screen. It looks like it's up to date in the most recent version. It's building. So I'll just quickly check the vignette. Okay, well, it's still in the vignette. So yeah, it's still referenced quite heavily. And I'm just going to check the reference real quick. Yeah, it's still in the reference. Okay, so it seems that it's still in the reference, so it's still a function within the current build of the subread. So once I finish, we'll have an investigate, okay, and see what's going on. But it's a relatively newish function, so it'll be weird if they got rid of it. Okay, so what we want to be able to do is read in all these transcript files from Salmon. And so I could just read it in using read.lim. Okay, it's basically a data table. This quant SF file is basically a data table. You can read it in. We did that in the end of last session, and it has all the transcript information. And so if we do this, to be able to get to this kind of gene summary. So yeah, there's a big caveat when working with Salmon. And so what we did with Salmon is a transcript level quantification. So it quantifies on a transcript level. So it's per transcript as opposed to per gene. And what we're doing here is differential gene expression analysis. And so if you have two transcripts that are part of the same gene, two different isoforms, they need to be summed up. So we need to be able to get a transcript to gene map. And that is one of the important steps in doing your TX import. Okay, and so to be able to do this, what we do is we'll just read in one of the files. Okay, so we do read.dlim. We read in one of the files of the quant SF files, and we just call it temp because we're just going to use it for a little bit, but we're not using it to read in important information. And then once we've done that, we can use the select function. Okay, and so what those transcripts encode for, what those these names encode for, and their equivalent gene is stored within these databases on Bioconductor. We built the index using Bioconductor, right? And so this came from Bioconductor, and it specifically came from this TXDB object. And so what we can do is we can provide it with this transcript names and say, I want to look inside this database and I want to get back every single gene that's associated with this transcript I'm giving you. And so the way this works is you use the select function. And the select is a key, like I use it all the time. We'll show another use later on, which is really important. And it's a key accessor for accessing these databases. And so you give it the database that you care about, which is the TXDB. You tell it what are the keys. So this is what is your search information. What am I putting in? And in this case, I'm putting in temp1. So we're giving in the transcript information. And we tell it this, I'm telling you, put in, I want, this is transcript name information that I'm giving you, and I want to get back these columns. So I want to get back the transcript name information, but more importantly, I want to get the gene ID information back. And so when you do this, you get this object we're assigning out to transcript to gene. And if we look at the first 10 rows, you can see we have transcript name information and gene ID information. So now we can map this transcript in this result from Salmon to what gene it belongs to. And then we can add all the transcripts together for a gene, and we'll have an idea of how many counts we have per gene, which we can then put into Dsq. So with that mapping, we can now do TX import. So we have to give TX import the paths to all of our quant_sf files. And so to get the paths, you can use this dir function. So we used the dir earlier, and it shows you all the folders in a directory, essentially, or all the files in a directory. And so we specifically, the way Salmon outputs is for every sample you count, it creates a folder, and in that folder is a quant.sf file. Okay, so we want to look inside every folder within this data Salmon, and we want to find everything that is a quant.sf. And so I'll show you how this looks because it's a little confusing. So I ran it. And so within this, we have these 5 different folders: treg1 quant, treg2 quant, and in each case, there is a quant.sf file inside them. Okay. And so by running this, it recursively looked through everything within data/Salmon, and it found every folder that contains a quant.sf file. And we also specify we want the full names because we want the full path. Okay. And so then we can provide these directories. We can tell it it's Salmon, and we can give it the transcript to gene conversion. Okay, yeah. Sorry if I didn't. So Sang just posted, our subread is part of subjunct. Sorry, I didn't make that clear. Yeah, our subread has a multiple algorithms, and one of them is called subjunct, and that is the one that is splice aware. So that means it's good for RNA transcription analysis. So yeah, once you get in our subread, you'll then be able to do subjunct. Yeah, so when we do this, we're importing and we're saving it to a Salmon counts variable. And so if we look at the result of when we do this import, you'll have it's a list and there's 2 slots in it. The first one is abundance and the second one is counts. And so the abundance is a TPM score, and the counts is the estimated counts. Okay, and if we remember, they're slightly different. TPM accounts for read depth, and it also accounts for gene length. Counts doesn't. Okay, but DESeq2 likes just counts. It doesn't want things that have been transformed or changed. It wants to make the models itself. So what we can do is build. There's a new operator. So if we remember, we used DESeq dataset from matrix or DESeq dataset function before. There's one that is specially built that imports from TX import. So we provide a DESeq dataset from TX import. We say that we want to give it the Salmon counts, which is our TX import object. And then we can just provide it the same metadata and the same group information we did before for the previous time. And it works fine. Okay, and then once it's a DDS, okay, so we actually, one point to note, we call this DDS Salmon. Okay, still a DDS object, we're just giving it a different name. We can run through the DESeq operation. We can run results to do the comparison. We can do again the same ordering, and largely it's going to be a very similar result. And so if we were to take if we were to compare the difference between the two following salmon quantification or R subregions hemorrhoids overlaps, if we compare genes that are p-adjusted 0.05 and to have a log twofold change greater than 2, that's an absolute log twofold change. You can see that we have about 1,200 genes that are changing in that way, and 930 are overlapping. And while we have about 300 which are either specific to summarize overlaps or specific to salmon. So it's not perfect, it's not exactly overlapping, but the core trends show that there's a lot of consistency between these approaches. So again, if you don't care about looking at the tracks on IGV, I do recommend doing that because it is an important quality control step. But if that's something less important to you, and you want to save time, you have lower memory, then you can skip the alignment and go straight to doing pseudo alignment. Okay, we're almost done. I just wanted to add a quick little thing about adding annotation, because everything we've been working on so far with corresponding to the genes is entree IDs, these gene IDs, which is cool, but actually is very not human friendly, it's very computer friendly. But we are all humans and we like to be able to read and understand what the genes mean, because there is information in a gene name. And so we can do a similar approach to what we did with select when we built our transcript to gene ID to be able to get the symbols, okay, and tons of other information within the object org.mm.eg.db. So this is the mouse specific organism annotation database. This contains information like Go terms. It contains information like ensemble IDs. It contains all the kind of annotation information associated with individual genes. And so we can say, I want to do select on this database. I want to do the row names from my result. And I know these row names are entree IDs, and I want to get back symbols. And when I do this, it gives me the symbol information. I can then, once I have that as a data frame, I have this as a data frame, and then I have my original data frame with the result. I can then just do a merge to put them together. So here is my entree ID to symbol. Here's my res as a DF. And what merge does is it looks for matches between the two and it sticks them together. So as long as you have a column in common, it can stitch everything together. And so in this case, I say by.x. That means I want the column that we're interested in is column 1, which is this one. It's the entree ID. By.y so this is your x here. For your y, we know this is the row names. So row names is 0. And then do we want to keep everything? Okay. What we want to do is, if there's, say you have a gene ID which is in this object, which isn't found in this object, do you want to keep it as an overhang where you have like some of the information from your E to sim, but there's no information in your result? Probably not. So you say all.x is false, but we want to keep every single result we got, even if they couldn't find the name of the gene in the database. So that's true. And so then with this like simple annotation steps, we've added in the symbol information into our result. Okay, and so we can see the top hits are Li6A, CCR2, GZMB. Okay, so that is what we're covering today. Again, apologize for overrunning. If anyone has further questions, I know we kind of sped through some of that at the end. Feel free to ask me the questions. I'll be here. I am going to stick around for half an hour, and then I'm going to go through some of these answers. So I would encourage, like this is where things start to get kind of a little bit more fun for some of the exercises, because you can load in these counts, you can start to do all these things yourself, and regardless of your setup, it should be able to run okay, and it won't take too long to do. And mostly what we're asking you to do is just do some DESeq analysis on a slightly different data set here. Okay. So yeah, if you have the time, I'd encourage you to do it, give it a go. It's the best way to learn. But if anyone has any other questions beyond that, feel free to let me know. And then if you do disappear, tomorrow we'll start at 1. It'll be the same structure, but as I mentioned earlier, what we'll be covering is in the morning, or the earlier session, we'll be looking at visualizing results. So we'll look at things like heat maps, we'll look at PCA, and we'll look at clustering. And then we'll do functional analysis. So that's gene set testing and things like that. So if you're only interested in specific aspects of that, you can also just dip in on that. Okay. Right, I'm going to stop sharing. So we do have a question. So it's related to this plot here. Charles asks, would the percentage of overlap between salmon and R subread increase if I chose a lower p-adjusted value? And so by lower, so this is one of the confusing things, like by lower, do you mean lower stringency or lower value as in 0.001? Where it's technically lower, but yeah, I think lower value. I think it would depend on your data set. More stringent, yeah. So if you did a more stringent value, it varies per data set. Typically, in my experience, yeah, like the sharp end of your analysis, where you get the really big changes, like if you were to do like a log 2 fold change cutoff and adjust your p-value cutoff, your data like between methods should be like very robust in that section. And in my experience, it is. It gets more and more like that. But it isn't always the case, and sometimes there's like very specific things that change it. And this, especially when it gets to, it all depends on your experimental design. And I think this also comes back to why we encourage people to work with us. We present here like a best case use that works for most of the time and shouldn't really throw up any artifacts. But you may have a specific thing you're interested in that this isn't the best tool for. And so one go-to, for example, would be some people like to use different aligners that aren't, say, R subread, that are more focused on splicing. And actually, I removed one of the slides. Let me see. On the slide real quick. But basically, there's some aligners work very, very permissively when it comes to, they're much better at finding splicing events, right? And so that's really great. Like if you've got this cryptic splicing, that can be super, super great. And some aligners are really good at finding that. However, that doesn't necessarily always bode well. And sometimes if you just have like a regular data set, you do these steps, and then yeah. So one of the aligners I'm really alluding to is star. Star is really good at finding isoforms. It's really good at finding splice sites, but depending on how you have it set up, it can be greedy. And so in this case, I have a zoom out of IGV, and everything that we believe is real is happening here. This is where the gene is, and we think this is real. Okay, so this is our subread, and then when we run it through star, it's picking up all these reads that are spanning all the way over here. And it's really hard to see, I know, because it's such a zoom out. But you can trust me, this is crossing like hundreds of kilobases. for these splice junctions. And so depending on your tools and depending on your setup, your very high value genes may change if you have something fundamentally different, such as something like this. That can fundamentally swing how much is considered to be part of a gene. And then if you're changing that, you can get massive amounts of counts, which can then completely change your counting. That said, I think for salmon and our subbreed, they're both relatively middle of the road, conservative, and just do a reasonable job all of the time. So for what your day-to-day uses, it should be fine. But yeah, if you're doing anything else or different, come and have a meeting with us, and we'll talk it out, and maybe we can help, or maybe we can just point you in the right direction. Does anyone else have any questions? Oh, okay, I'm going to pause recording for now. Start recording again. Okay, so everyone should be able to see my screen. And so that's good. So what we're doing in these exercises is we're actually just we're going through and we're doing a differential analysis, and we have a different data set here. And so instead of considering Treg cells, we have some tissue specific data. We have liver and heart. And this is actually a data set we're going to keep on using. In fact, the start of tomorrow, we're going to focus on this data set because it's a nice complex data set. And so there's liver and heart here, and we've already done the counts for you. So that simplifies things. So first step, I'm going to load in these packages. We have our annotation package, we have an import package, and we have DESeq2. And so what we're asking is to get the counts into DESeq2. So we want you to load the data, create a DESeq2 object, and then we want you to actually visualize it. And so because it's a log2, the visualization, you need to add something to it, because you can't, otherwise you'll get all these infinite values, and it will get upset when you try and do the logarithm because of the zeros. So we add 0.25 standard amount to everything, just so we can get a visualization. And so we load in these packages. We're actually not using all of these packages right now, but we need some of them. And so I'm just going to load this in. So this is gene counts tissue. Okay, and so this is a range summarized experiment that we've met before. And we can actually, you'll notice that the cold data has nothing in it. Okay, so hopefully everyone was able to intuit that sorted heart 1, sorted heart 2, and sorted liver 1, sorted liver 2, they correspond to heart and liver. So we can add cold data that, oh no, I don't want to do this again. We can add some column data to that. So why are you being so sensitive? There we go. So let's do, we can access the cold data of this, the cold data accessor function. Okay, we have none available, so we're going to assign some in. So I'm just going to write it out. If you have more complicated data set with more samples, you're going to want to find a way to do this computationally. But we're just going to do writing because it's easy. What did I do wrong? Oh, I know why. So I create this factor vector. Okay, that's heart, liver, heart, liver, liver. It has 2 levels, heart and liver. And I've assigned it specifically, I've assigned it into the coal data, but specifically I created a column called tissue and assigned it into that. Okay, so now when I look at coal data, like before I got this as the answer, now I have a column called tissue, it's a factor, and I have this information and it's associated with the correct samples. Nice and straightforward. Okay. And so you may have done a more complicated one, which is totally fine. You create a separate data frame and then assign that whole data frame. I'm just showing you some nice little shortcuts, especially when you're working with a smaller data set, so you don't have to do so much. So next, because it has all the row ranges, it has everything associated with it, all we have to do is do Dsq data set on it. Okay, and now we have DDST, and we have this DEseq data set. Okay, and this is our tissue data set. You can see it has some metadata, it has our assay information, everything is contained within it. And so then last gets to this visualization, which is different to what we've done so far. And so there's lots of ways you could do this. Some people are ggplot people, some people are beta people. If you don't know what that means, it doesn't matter. But we just used boxplot, the boxplot function, and I'm supplying the counts. And I didn't want the normalized counts, I want the unnormalized counts. So I say normalized equals false. But we didn't actually specify. So if you want to do normalized equals true, you can. We did plus 0.25, and then we did log2 of the whole thing. Okay, so this is our data input right here. Then we just add some names. So heart 1, heart 2, level 1, level 2. So these are some nice names to be in the axis. And then we're also adding a color. Okay, so we have 4. So this is brewer.pal to get color. And we say we have 4 entries and we want to use the paired palette because we have paired data set. You don't have to make it look so nice, but I think it looks better this way. And so you can see that broadly they're the same. The median is very similar. There is some difference in heart 1 in the variation, and maybe we'll see what that means tomorrow. I cannot remember off the top of my head, but maybe this one will stick out more than the other ones when we do some things like PCA and things like that. But yeah. Okay, well, so now we want to do exactly the same again with the normal. Oh, yes, yeah. So at the moment, we hadn't done DEseq. So I don't believe if I did this again, it's probably going to give me an error. Right, and there is an error because we haven't performed DEseq. So at the moment, the DEseq object, the DEseq dataset, DDS, whatever you want to call it, it has all the information associated with your data set, but it hasn't done anything to it yet. It's just a container, you put it all in, but that container hasn't performed any operations. So it hasn't done the normalization, it hasn't done like variance shrinking and all that stuff. And so to do that, we need to do the DEseq. Okay. And so, yeah, we always assign it back to itself because it still contains all the same information. It just has more information once it's performed DEseq. And so now, if I was to do counts DST normalized, no. Maybe we'll just look at the head on this. So we just look at the top. Now that works. Before it wouldn't have worked. Okay, and if we can compare it to normalized equals false, so you can see the difference. Right? We expect normalized is false. They're all going to be integers because that's the count information we gave it in. And now it's been normalized. And so you can see that, say, some of these have been scaled up. This has been scaled down. This has been scaled up. This has been scaled up. Okay. And so now we can just draw exactly the same box plot. Now it's with the normalized values. And it looks very similar. We do this. There's some slight shift. This is shifted down a little bit, so it's more in register with everything else. And these two were a little bit lower, and now they're up a bit. Okay, so as a global level, not much has changed, but there's some subtle hints as to the relationship between your data sets. Okay, next we're going to just do dispersion estimates. Okay. And so you can see really nice and clearly with this data set the relationship between very low counts and dispersion. And that actually becomes less strong, especially once you're over like 100 counts. And then it's like very like on a level. And so we get this shrinkage in both directions towards the fit. And these ones are not shrunk because they are they're too far away from the fit to be considered. Okay, so next step is we're actually going to do the differential analysis, and we want to add a p-adjusted value for all genes, and not just the ones that Dsq tests. Okay, so we use ggplot for this, but the key thing here that we're digging into is this concept that DESeq doesn't test every single gene. It realizes that a lot of these genes we don't care about, and it doesn't test them. Instead, it gives them an NA. And you can see why, right? Like all of these ones at the end, they have no counts associated with them. So why would you try and do adjusted p-values with them? And it just penalizes your testing. And you can see here, like this base mean, and the average count per sample is 1.5. So it's not worth just even considering this for differential analysis. And so it doesn't consider it for multiple testing, and that gives you more power. Yeah. Okay, so to compare the two, we'll do, we have to convert it first to a data frame. So at the moment, it's a results object. Right. And so nothing much is changing. Right, it's the same data, except this one is a data frame. And so it's more accessible to other R functions, which we need when we want to do a p-adjust. So p-adjust is it's a p-adjust function. Yeah. So we're doing the p-adjust function, and we're doing it on the p-values, and we want to do Benjamini-Hochberg. Okay. And so this function is not part of DESeq. It's more just a generalized p-value function. But we can use it on the results that we have. But it doesn't work with a DESeq object. So that's why we converted it out to the data frame. And so now we assign that to a new value, which is new p-adjusted. Okay. And so again, you can see this top one is an NA because it's really low. And now we're considering it's a 0.83, right? And interestingly, you can also see this one is a significant value, 0.01. And when we include everything, it has a 0.013. Okay, so it's not made much difference. It's 1,000th of it's gone up by 3 1,000ths. No, yeah, 3 1,000ths in its p-value score. But that it could make a difference to your numbers when you're considering 20,000 genes or whatever the number is. And so we're now asking, this is a complicated if-else statement. Okay, we're saying if-else, so is the new res df less than 0.05, and is the log twofold change greater than 0? Okay, else is it less than 0.05, and is it less than 0? Okay, and so using these if-else statements, we're labeling whether in the new P adjusted, whether it's considered to be a heart up, heart down, or no change, based on whether it meets these conditional statements. So if you're super new to R, you're struggling with some of this, don't worry. This is like some more intense R stuff. This is an opportunity for you to learn some basics of R. And so like things like if-else statements and things like that. And don't worry if it just doesn't make sense. Okay, you can always revisit the intro to R section, and that will explain everything. Okay, and so I do exactly the same thing, but now I'm considering just the original adjusted p-values and not the ones we created ourselves. And so this now we have two vectors, and one of them tells us, is it considered significantly changed in each data set, and whether it's heart down or heart up. Okay, so then we create two data frames with this value. Okay, and so and what data frame? There you go. And so what you have here, if you compare the two, oh, that should be interesting. Oh, it's because it's an NA. Yeah. Yeah, what you have here now is everything labeled for every single gene, whether it's a heart up, heart down, or no change. And then the NA values just stay NA. And then we can stick these together. And when we stick these together, it's just these two data frames stuck together end on end using rbind. And the important part of this is by doing that, we can then, it's one data frame, so we can actually use it for ggplot. And so what ggplot does is we can then say we want to consider on the x-axis the adjusted p-value, and we want the fill to be method. And what it's doing is it's just counting how many fit into each group. Okay, so how many times do we see heart up? How many times do we see heart down? How many times do we see no change? And how many times do we see NA? And so the important thing with this is you can actually see, despite the fact that we're filtering out all these genes because we don't think they're important, we get in the filtered data set, we get more genes that are considered significantly down, more genes that are considered significantly up, and less genes that are considered no change. Okay, and a large proportion of that no change is actually going towards NA values, but also a large proportion, a small proportion is going to these values. So we're actually, the difference between here and here is why we do that filtering step beforehand. Okay, so it's small stats, but these incremental gains are really important for getting as many genes as you can. Okay. Right, next question, we're almost there, is producing NA plots. Okay, so to look at it before and after this shrinkage we talked about. And so the shrinkage can be really important if you want to do things, as I mentioned, like GSEA. Typically, you can use other values that are the result of DESeq2. You can use the adjusted p-value or the stat value as your ranking for GSEA. And we'll get into why that's important during the GSEA section of the course tomorrow. But quite often people like to use log twofold change, because then you can also consider both the amplitude of change as well as the significance of change. well, they like to consider the amplitude of change, but by doing this shrinkage, you can actually consider amplitude and significance at the same time by downweighting the ones that are like only like they have a big change, but they're not very significant. So first step is we actually need to do our differential again. We already had that. I didn't really need to redo it, but hey. And then there's the plot MA function. Super easy. It's really nice. There's a bunch of just plotting functions that are built into Dsq. And so then for the LSC shrink, there's an LSC shrink function. You provide it with a DDS. Okay. And so it tells you about the function it's using for this, gives you a reference. It takes a sec. So you can actually see. And so actually when we ran this before, we actually provided the coefficient name, but in this case we're telling it which coefficient we care about. And I think you can, is it does this work? Oh no, oh no. Yeah, and so these are the two coefficients available in the DDS object. You have the intercept and you have this one, okay? And so we care about comparing liver versus heart. And so that's why we're saying the coefficient too. So if you have multiple groups, lots of things going on with your object, you can specify more specific explicitly. But for what we're doing, this is totally fine. And then the final thing is plotting the MA again. Okay, so you can compare the two. You can see the difference in shapes. Okay. You can see the penalization down here. Most importantly, like these are not significant genes, and they have high log fold changes. And so if you were to base it on the GSEA, like these genes up here, which are very insignificant, would be higher ranked than a bunch of your like much more interesting genes. So by doing this, then it's much less of an issue. Okay, so that is us. That is the exercises. Thank you to a few of you who stuck around to the end. I hope that all made sense. If it didn't, you feel free to let me know and I can clarify things. And I look forward to seeing, where is that? Ooh, there we are. Yeah, if that didn't make sense, yeah, let me know. We can also discuss things tomorrow at tomorrow's class. Okay. Right, I'm going to stop recording.